Thank you very much indeed for the kind invitation to come and talk to you today. And you mentioned about the importance of you talking amongst yourselves and the experts being left in the corner. Believe you me, the experts are very keen to hear from you too. Well, this expert certainly is, because the sort of reward that I get from meeting you is the encouragement to carry on with our research, because you get a lot of inspiration of meeting patients and relatives of patients, and you go back and work that little bit harder to try and discover new cures or discover causes of various tumour types. So it's a real privilege to talk to you today. At the end of the session, I hope there's going to be a chance for some questions and answers. You come up with the questions and I'm meant to come up with the answers. We shall see. I'm sure the questions will be very perceptive. The answers may not be so. It's nice to talk in this particular place, because I organised a conference here once, and one of our delegates made the mistake of asking for a taxi from the station to the hotel. and he got in the taxi, and the taxi driver said, "Wait, man, if you get on the other side, you'll be there." And very kindly didn't charge him for that service. It is incredibly convenient. I'd also like to reassure you that Newcastle is not always as damp as this, but we have plenty of water, so those of you who are further south, we are willing to negotiate a price. Okay, so with no further ado, my brief today is to talk to you about tumour biobanking, which for a group like you, I know is something that you're particularly interested in. But before I start, I want to explain my multiple roles. I'm a complicated person in the sense if someone said, "What do I do?" Well, I do lots of different things, hopefully moderately competently. I wear many hats. So originally I'm a haematologist, a clinically qualified haematologist looking after patients with leukemia, a very different sort of disease to the one that you're familiar with, but with commonalities, and I'll come on to that when I talk about the research that we're doing in Newcastle. I'm also a researcher, been doing this for 25 years. I've enjoyed every minute of it. It's exciting and very rewarding. I don't even call it a job, really. I'm a certain sort of age. I've got white hair and a white beard, so therefore I'm senior. I happen to now be the director of the Northern Institute for Cancer Research, which is the poor northern relation of the Institute of Cancer Research that you're probably familiar with in London. And we do a similar sort of activity. Our particular thing in Newcastle is, like the Marsden Hospital, trying to develop new drugs to improve the outcome of a multiple number of patients, both children and adults, with cancer. I'm also very privileged to be the so-called designated individual under the Human Tissue Act. Okay, I've decided to spare you a 45-minute exposition of the Human Tissue Act, although it does influence what we do quite significantly. And I see my role in that particular function is to facilitate research wherever possible, and to do that I try and take as much of the responsibility for what we do on my shoulders as I can. I'm also the director of a biobank in Newcastle University, which has a multitude of different samples from a whole range of patients with cancer. We've got about 30,000 samples in our bank at the moment. And last, I am also the pathology lead for the Confederation of Cancer Biobanks. So what this is a loose affiliation of like-minded individuals who want to promote the collection, storage and use of human tissues for research purposes. So I think possibly it's the latter role in which I'm speaking to you today, but not ignoring all the others. So briefly about the Federation. It's, as it says here, a network consortium of independent biobanks. There's no central organisation which funds, regulates or supports biobanking activity in this country. And the idea is really to share best practice and to represent those biobanks and promote their activity, and to try and move them out of the shadows which sometimes they are consigned to make sure that we set up good quality biobanks to further our research aims. So it offers networking opportunities for like-minded researchers. It's very keen to promote interactions with both patients, relatives of patients and the general public, to make sure that biobanking can be promoted, and having a collective voice hopefully pushes that forward with the opinion formers and politicians of the country. Okay, I thought also I'd start with a few basic terms. Now my guess is that most of you here are actually in your own right little experts on your particular disease. You normally do find that people know an awful lot more about their particular disease than somebody who's meant to know about all of them often does. So I don't wish to insult your intelligence, but sometimes it's useful just to get some basic terms out of the way. And first, of course, is cancer. So to my mind, cancer is the unregulated proliferation of cells that have lost the ability to behave properly. Okay, so they divide when they shouldn't and they don't die when they should. And in a nutshell, that's what cancer is to me. As you will know, it's considered now to be a genetic disease, not in the sense that it's inherited from parents to children, but it's caused by damage to the coding of the cell, the DNA. So our main aim is to discover ways in which we can understand those abnormalities of DNA and target new drugs to be able to be selective against cancer cells and leave normal cells relatively unmolested. So this is a fairly state-of-the-art CAT scan showing a gastrointestinal tumour living inside the stomach of this particular patient, and basically it's a lump, okay? And you'll all be familiar with that terminology, I'm sure. What about tissue and biopsy? Again, I'm sure you do understand this, but it's surprising how often members of the public don't understand what we're talking about when we talk about biopsies and tissues. So biopsies is, to my mind, samples taken from tumours. They can be taken at the time of operation, or they can be taken using needles and other processes when the patient's awake and not fully opened up for a surgical procedure. And tissues are simply sections of that sample, often placed on microscope slides to be analysed. And this gentleman here is a typical microscopist. This is what we all look like in the laboratory when we're toiling away. And what's he looking at? He's looking at something that looks a bit like this. So I'm sure you'll be familiar with this sort of picture. So these are the misbehaving cells I was referring to before. Again, it's your particular tumour type. These are described as spindle cells because they're long and thin. So the blue things are the nuclei of the cells, the red bits are the cytoplasm of the cells, the surrounding nutrients, etc. And the DNA lives in the blue bit in the nucleus, and that's where the damage has occurred. Okay, so we're all on the same page with regard to biobanking. So what can you do with this sort of thing? You can also store it, and this is what a biobank is. So a biobank is just a collection of these tissues. They can vary in size. You can actually have a bank with whole organs in it. A little bit of a challenge storing large numbers of those, and also of less value than actually large numbers of patient samples from different patients, rather than a large amount of material from the same patient. And you can store them in tubes, and we normally store these things at very cold temperatures, simply to preserve them for long periods of time. And so they can be non-viable, in other words the cells are dead, or the cells can be disaggregated, split apart, put into a nutrient broth and kept viable, and then turned into something called cell lines, which divide inside the laboratory and are fantastically useful for our research if, for example, we're testing new drugs. So the best sort of sample, if you can get it, is viable material from a tumour. Now to do that, of course, you have to link up with your surgeon, you have to link up with the whole team that are looking after you during an operation, make sure somebody's on hand to be able to take the sample, break it into parts, put it into the broth and take it to the laboratory. All of those things sound fairly straightforward. All of them are fraught with complexity and risk within organisations which are not set up specifically to do research. The organisations we're dealing with is the health service, which is set up to make you better. Okay? So a lot of what we do in biobanking is incredibly mundane, pragmatic, practical, and deeply frustrating, both for us, and I know, having already had some conversations about this, to you, in the sense that why can't we do something which is relatively straightforward in an expedient way? And it's all to do with organisations. So I spend a lot of my time, A, talking about regulation, and B, talking about getting the right person in the right place to do the right thing when it comes to biobanking. Okay. Not the most exciting of jobs, you might say, but ultimately the most rewarding. Okay, so why is it important to collect this material? What's all this tissue for? And I thought really I would take a brief canter through the sort of research that you can do using material from patients. And one thing that 25 years worth of research has taught me is that you do need to go back to patient material as often as you can. If you rely on artificial model systems, such as in rats or mice or other animals, you learn a lot about cancer in rats and mice and other animals. You don't necessarily, which is good news for them, but you don't necessarily learn an awful lot about how to treat cancer in patients. So you need to all the time refer back to patients to make sure that the models you're using are accurate. And one of the things we find, for example, is that the cell lines that you can develop from patients are very useful, but sometimes they're quite atypical because they develop into a cell line. Now, if I took 100 patients with a GIST and tried to develop a cell line, probably only about 5 or 10 of those cases would happen. So there's probably something about those 5 or 10 which make them into cell lines in the first place. So if you find something in those cell lines, you have to go back to the patients to make sure how representative it is. So getting samples from people is incredibly important. Why is cancer research important? Well, 1 in 3 people will develop cancer at some stage during their life. And research basically can be into causes or cures. And our emphasis in Newcastle is on cures. So I thought just a brief outline of what I wanted to cover in this section of the talk. Basically it's a little bit about how cancer arises, and then a bit about how you develop new drugs. I hope that would be interesting to you today. Okay, so this is a picture of widely metastasized cancers in the liver and in the lung, and in the white bits are the cancerous bits, and the red bits are the normal bits. And you can see that there's widespread metastasis. Metastasis is spread of cancer throughout the body, away from its primary site. And once a patient has got these metastases, it becomes quite difficult to treat them. And obviously surgery in these sort of people is very difficult, because getting each of those individual lumps out is almost impossible. And in fact, I've got a pond at home. Many of you will have a pond, and I've got a blight of duckweed in it. Right? And every time I look at that duckweed, sadly, for the sort of person I am, I think that that's what we're trying to do with cancer sometimes. Getting rid of that last blooming duckweed. You think you've got it all out, don't you? And you come back in a few weeks' time, and it's grown back again. So you can't, unfortunately, put chemicals into a pond because it will kill all the fish. So the mechanical approach is all you've got on for you. Whereas for patients, what you're looking for are magic bullets which will just kill the cancer, but not kill the normal cells. And therein lies the problem, because cancer cells and normal cells are actually very, very similar. So we've been very successful in curing infectious diseases due to bacteria, for example, but a bacterium and a normal cell are very different. So it's finding that Achilles heel, that difference between a cancer cell and a normal cell, which has been the problem that has blighted us for the last 50 years or so. Okay, so what's cancer caused by? I referred to this very briefly just a second ago. And it's a balance between cell division and cell death. Okay? And it's only recently that we've understood that the cell death side of the equation is important. When I first started cancer research, nobody talked about the fact that cancer cells didn't die when they should do. As part of the regulation of our normal body, cells die routinely as we grow. So, for example, in your gastrointestinal system, cells are regularly dying all the time. It's a normal process, and it's lost in cancer. So that side of the equation, which really wasn't understood, the unregulated cell division has been understood for as long as people have been describing cancer. We have the most enormous number of cells in our body. A colleague of mine tells me it's 100 trillion. I don't know if he's counted them himself. And the thing is, they almost all of them are dividing much of the time. Every time they divide, they double the amount of DNA in the cells. DNA's got 30 billion bases in it, the code, and every time it copies itself out, errors are introduced. Inevitably, with that amount of copying work, what system wouldn't generate errors? And so the miracle really is that cancer doesn't occur more commonly. We've got all these cells dividing. Every time it divides, the DNA has to go through this fantastic trick of replicating itself, copying out. Remember, you know, I don't know about you, but if you copy something out, you almost make mistakes, don't you? And it's the same in the human body. If we didn't make mistakes, actually, when cells divided, we'd all be amoeba. And you wouldn't be listening to me now, which might be a relief to you, I don't know. Because we need those errors to be able to evolve. So it's part of the evolutionary process. We have mechanisms to try and reduce the error, but they're not 100% effective. And cancer arises because of mutations which occurred. And it's down to the inspirational thought of this guy called Alfred Knudsen, who looks like a sort of slightly exasperated librarian to me. Looks like somebody who's you know, disappointed in some sort of way. But his idea was that you needed more than one mutation, more than one abnormality in your DNA for cancer to occur. So, for example, if you take 100 children at birth and take a sample of their cord blood, 1 in 100 of them, 1 in 100, which is not that infrequent, will have a mutation in their blood cells, which if it's met by another mutation, which occurs again with a frequency of 1 in 100, will develop cancer. So it's the fact that 2 or 3 mutations happening at the same time are required for cancer to occur, which probably explains why it's not as common as I initially said. So Knudsen pointed out these mutations occurred in a clone of cells which was pre-malignant and then occurred again to form something that was definitely cancer. Okay. So that, in a nutshell, is where we now understand how cancer develops. How about developing some treatments? How about how we approach this? We've approached it in a number of ways throughout history, and we're still working out the best way of approaching it. And the first way is just by simple chance observation. And chance observation in science generally are really important. And this is the sort of work that we've been doing in Newcastle, drug development, in the laboratories up at the Northern Institute of Cancer Research, where I come from. And as I said before, it's all about developing new therapies. So go to the chance observations. I like this cartoon. I feel very similar to that sometimes. I'm sure we've all been pointed out the errors of our ways by our lives. So the first chance observation I want to talk to you about came out of the tragedy and misery of the First World War. So this is a picture of troops in the First World War. If you look carefully, they've got bandages over their eyes, and they're being led in a trail because they're all blind. Okay? They're blind because they've been exposed to war gas during the First World War. And one of the drugs, one of the agents which was used during the First World War was something called mustard gas, which is a truly evil invention. And it's a very simple molecule. It's got a sulfur in the middle and these 2 chlorine atoms. It's an incredibly reactive substance, okay? And the chlorine atoms basically fall off very easily. And it's almost a gas. It's sort of oily liquid. And it dries out the skin, causes burns, and if it gets in the eyes, it causes blindness. And it used to sort of waft across the trenches. Horrible. But it was also noted that, as well as doing that, it caused the blood cell count to be reduced. And somebody said, oh, well, if that does that, what about treating blood diseases where we've got too many cells? What about giving it to patients with cancer? And the initial papers talk about mustard gas itself being used to treat patients, and it was used for skin cancer. It was dropped from a dropper onto a skin cancer, and the papers talk about the need to have the window open when you were doing it. because the gas was so toxic. So some chemists decided to modify. Oh, this is a picture of what it does. So this is what it does to your foot, this horrible blister. This is a white cell on the right-hand side to remind me that that's the white cell count went down. So imagine that. Please don't imagine it too long in your eye. Okay. So what they did, they substituted these the sulfur bit for a nitrogen bit, which means it hangs on to the chlorines a bit more. That's my understanding of chemistry, which means it's not quite as reactive. And that drug, nitrogen mustard, is an anti-cancer drug which is still used, and actually was developed down at the Marsden Hospital originally when it was the Chester Beatty Laboratories. So that's an example of chance observations. Now here's a nice job if you can get it. This is a picture of a range of natural products. So there's also been a long history of people finding anti-cancer drugs in nature. And there's a gentleman called Pettit who works on the Californian coast, and he spends his time scuba diving. Where did I go wrong? He spends his time scuba diving, looking for exotic organisms from the wild, crushing them up and seeing whether they have anti-cancer properties. And as I say, that's been very effective. Most recently, the Pacific yew tree was found to contain a very potent anti-cancer drug called Taxol. And the example I want to give today is this picture of the May apple, which is apparently quite a common plant. And if you crush that up and purify the contents of your test tube, you'll come across something called etoposide, which is a drug which is used routinely in the treatment of many cancers, and in my particular field, in childhood cancer. And it's been described way back to the Leech Book of the Bald, in 950. So in other words, ancient cultures knew that there were these agents out there which were effective. So there's still a lot of effort put into discovering new anti-cancer drugs from natural products. And there's a vigorous debate about who that intellectual property belongs to, interestingly. Okay, but we like to live in a rational age. We like to think that all this science that we've supported over the years means that we can get away from crushing things up and hoping that chances will come our way. And we want to have a rational drug design, using the science that we've discovered over the last 50 years and putting it into action. And many of the drugs that you'll be aware of have come by that route. But in fact, this started quite a long time ago, and everybody has to have a pin-up, a hero and a heroine. My heroine is this lady, Gertrude, shown with Hitchens, who was the person that she did her work with. And when I was a very young haematologist, I went to one of the huge conferences they have in America dedicated towards latest research advances in my own speciality, haematology. And they have these enormous halls where you have posters where you stand by and people come and ask you questions, and you gain a lot from discussion. And I was standing very proudly by my poster. My beard was very ginger then, and quite long. And in wandered this elderly lady. And I'm afraid to say, I thought that she'd come thinking it was a shopping mall. Right? So she wandered in, and then she started asking me the most perceptive questions. And luckily I realised in time it was the Nobel laureate, Gertrude. And she won the prize for discovering a drug called 6-thioguanine. She was working at a time in the 1950s when DNA, the structure of DNA was only just being described by Watson and Crick. Okay? But she, very early on, realised that DNA was going to be very important. Wasn't quite sure what it did at that time, believe it or not, but she thought it was important. So she designed drugs with Hitchens that interfered with the normal synthesis of DNA. Okay, so this is a picture here of the DNA, the very iconic double helix on your left-hand side. It's got four bases in it: adenine, thymine, guanine, and cytosine. Right? And what she did was, she said, I'm going to fool the cells, right? I'm going to catch them out of their own game. I'm going to devise a drug that looks incredibly like guanine, right? And the cell will incorporate that into the DNA, and the DNA will stop dividing. And it worked. It worked fantastically well. And 6-thioguanine and 6-mercaptopurine are still two drugs that are used all the time, for example, in the treatment of children with leukemia. I've not added it up, but if you can think of the number of children's lives that she's contributed to survival, it is enormous. So not somebody looking for the shopping mall, after all. Okay. So childhood leukemia is one of the great success stories within the anti-cancer world. There's about 350 cases in this country per year. Okay. And if you go back to the papers in the 1950s, you come across wonderful phrases that talk about it is totally immoral to treat any child with acute leukemia because everybody knows that they'll be dead within 6 months. Okay? Makes some salutary reading, actually. And the people who started to try and develop these new drugs, like Gertrude and others, were pilloried for their activity. They were accused of being terribly immoral because they were giving false hope when no hope was justified. And it's through the courage of people like her and others that challenge that. So if you hear someone say at the moment that, you know, this sort of cancer is never going to be curable, that sort of cancer is never going to be curable, just bear in mind Gertrude Alien, bear in mind childhood ALL. Okay. So it occurs, you know, quite commonly in children with a peak of around about 3 or 4. And acute lymphoblastic leukemia is the most common sort, and those little rather attractive cells for the nastiness they cause is a picture of what they look like. And there's a little bear in there who's obviously having a quite intensive treatment for their leukemia. And this is an old slide now, and it shows the relentless progress that occurred in leukemia during the 1950s and 60s. So here's 1950, there's Gertrude Alien and her colleagues, and then during the 1960s to the 1980s, a fantastic rate of improvement. What an exhilarating time to be involved in the treatment of patients with that disease, it must have been. Progress flattened off after that, and having said that, the cure rate in acute lymphoblastic leukemia is now about 85 to 90%. Okay. And you're left with a sort of a rump of people who are quite difficult to treat. And the treatment itself lasts 3 or 4 years, which is pretty tough if you're a child, and it's associated with some long-term toxicity. So the challenge is now in that disease are actually doing something about reducing those toxicities rather than all the time worrying about increasing survival rates. What a great position to be in. Okay. What about targeted therapies? That's our rational design. And this really owes much to the thinking of these two gentlemen, revered Victorians. Paul Ehrlich is famous for the coining the phrase the magic bullet. He actually worked on dyes. He was interested in how dyes selectively stain cells. So if you remember the slide I showed at the beginning with the blue blobs being the DNA and the red blobs being the cytoplasm, he said, well, if you could do that with drugs, wouldn't that be great? So he invented the concept of the magic bullet. And of course the gentleman on the right, your left, right, whatever, right, is Fleming, and he was the father of all chance discoveries, really. But the chance discovery goes with a prepared mind, as the phrase goes. He had the genius to recognise what he was seeing. And again, that was referring to bacteria. And that thinking informs the idea of targeted therapy. Hello. Yeah. Now, I want to, before I tell you the example, which you'll all be very familiar with, of targeted therapy, I want to introduce you to these couple of bouncers. Right? And these gents look like could work down the road in one of our dodgier clubs. This guy's called Hungerford and Knoll. Knoll's a good name for somebody in science, I think. And they were responsible for describing the cytogenetics of cancer cells. So cytogenetics is the study of chromosomes. Okay, your DNA is divided amongst a number of these thin threads called chromosomes. And a lot of our knowledge about cancer has come by an analysis of that. And they spent many a happy hour looking down the microscope at the little squiggles that are shown behind them. And their particular thing was that they were responsible for karyotyping patients with chronic myeloid leukemia. Okay, this is a picture of a karyotype. These are the chromosomes laid out in a neat row. They're not like this in the cell. So what you have to do is to take a picture once you've got the cell divided in a certain sort of way. And the old-fashioned way of doing it was to cut up the picture and line them up like this. And if someone opened the window when you were doing that, boy, were they unpopular, as your chromosomes went flying across the room. Okay, so this is a normal karyotype from a male, because it's got an X and a Y. Okay, and what Hungerford Knoll and somebody called Janet Rowley, I'll show you a picture of in a minute, were famous for discovering is the cytogenetic abnormality associated with chronic myeloid leukemia, which is in my neck of the woods as a leukemia. So CML, when I first started doing haematology, CML was not a very nice diagnosis to come up with. It usually presented to the ages of 30 to 50 with a very high white count. Sometimes the white count would be 10, 15, 20, 100 times more than it should be. The cells themselves were abnormal. There's a picture down there of them. And not only did you have too many of the wretched things, but they didn't do their job properly either. So you had too many white cells not working very well. So patients with leukemia present with bruising, they present with abnormalities of responses to infections, and generally pretty unwell. And you gave them some sort of Second World War type medication, hypoxanthine, hydroxyurea rather, and they were usually jolly sick and sadly normally died within about a year or two, okay, if you treated them. And that was before the fantastic discovery of the Philadelphia chromosome. So the Philadelphia chromosome, described in, you've guessed it, Philadelphia, is actually a translocation between two chromosomes. In other words, two chromosomes, 9 and 22, break and exchange material between the two of them. Okay, and this was really described by this wonderful lady, Janet Rowley. I was in America in December last year, and she was giving a seminar. She's still working. She's in upper 80s, and fantastically crisp. I mean, really, really crisp. Fantastically modest as well. So I had the privilege of being able to go to a session where there were just 15 or 20 people, mainly students, maybe young people, wanting to go and meet her. And she was so modest about her achievements and so inspirational about what she was doing, and still so keen to know what was going on. And she describes her career fitting in with a family life, and how, you know, how it was all, in her opinion, a fantastic accident that she managed to do the work that she did. I don't believe a word of it. You know, I believe the word about the family life, but not the fantastic accident. She must have been, and still is, incredibly, incredibly bright. And she described this chromosome, so it's got a helpful arrow next to it, and you can see it's a bit longer than it should be. So this exchange of material between two chromosomes has a very profound effect, which is deeply involved in the generation of chronic myeloid leukemia. Okay? So this is a cartoon representing the two chromosomes, and you can see the materials exchanged. So you get an incy-wincy teeny-weeny one, which is the Philadelphia chromosome actually, and a slightly longer one, which is the partner of those two. What this does is bring one gene on one chromosome next to another gene on another chromosome, and that has a very profound effect, because what it does is it turns the gene on. In particular, it turns on this gene ABL. Okay. Now, what ABL is something that drives cell division. So we've all got ABL in our cells, and normally it drives cell division, and we're quite happy. But it's like getting your foot stuck on the accelerator if you put it next to BCR, which is the gene from the other chromosome. So that sticks the foot on the accelerator. The cells are dividing when they shouldn't do, and far too often in an uncontrolled way. And if you can fix that foot on the accelerator in CML, you can practically cure the disease. Okay? That's unusual in cancer research, but it made a huge difference to patients with CML. So overnight, patients were demanding to go on imatinib, okay, which is a drug you'll have heard of in your context, because it's also found out to inhibit one or two other tyrosine kinases. So tyrosine kinases are the accelerators I've talked about. So the drug itself is not absolutely specific to BCR-ABL. It also affects genes turned on in some forms of GIST. And this is where I have to bow to your experience. I am told that imatinib is relatively easy to take. Now, that might be a relative judgment. I would bow to anybody who may wish to disagree with it. But compared with the nitrogen mustards, compared with the drugs that we're still left with treating a lot of patients in, the side effects are more manageable. I don't have to take it, so that's easy for me to say. Okay. Now, what's happened, of course, is some patients have developed resistance to this drug. Their tumours are very cunning, and they've discovered ways of getting around it. But still, imatinib remains the pin-up of the targeted therapy world, and one we all aspire to replicate in other disease types. So just to go over what it does in a little bit more detail, it actually works in the ATP sites. The ATP sites where molecules get their energy from, okay? And it sticks in the ATP site, as I say, and interferes with the process. And Novartis were the first company to exploit this, and they invested a lot of money and faith in the drug. There were times when they nearly pulled out. And the interesting thing is they were all told that they would never make any money out of it because there aren't many patients with CML. And for many years, that was the argument that stopped these drugs being developed. It's not true. They have made quite a lot of money out of it. And that's been good for the whole field, because it's meant that other companies have not been able to follow that argument that they're not going to make money out of it, because they will. So finally, I just wanted to sort of highlight where we are. This is a bit of an old slide now, but you can imagine this graph has continued to shoot out the ceiling. And this is the rate at which you can sequence DNA. Okay, so I've said several times now that cancer's a disease where DNA is abnormal. Wouldn't it be nice if we could sequence your entire DNA from your tumour and find out what abnormalities there were, and then target our therapy against it? What a dream. How could that ever possibly happen? It took 15 or 20 years to sequence the first human genome, and it cost multiple millions of pounds. Okay? You can now sequence a human genome for $5,000, and it takes you a week. So we are very close to a stage where you'll be able to sequence every individual's tumour and know what you're up against. And there's reckoned to be about 70 different genes that can be affected in cancer. What we now need is a suite of drugs to target those genes, to turn off their activities, to make the cancer go away. So the trouble in our area, we're always talking about, how it's going to happen tomorrow, it's going to happen tomorrow. But if you look at the underlying science, there's a real belief within the field that we are at the point where we can make real changes. This graph really shows, as I said, it's a bit old, it shows the number of DNAs that have been sequenced, and that's really shot up now, because as I said, it's something I could order. If I go back to the lab this afternoon, I could order, if I had $5,000 in my pocket, some sequencing to be done within a very short period of time. So there's a place called Future, apparently. Where next? And it really depends on the sequencing of the human genome, which occurred 2000. So I mean, it's now 12 years since the sequence was first obtained. The thing about the human genome sequence was that it was only the start, really, because it gave us the code for all the DNA, but it didn't tell us what all the genes did. And so we're still finding out lots and lots about the human genome and how we can apply it to developing anti-cancer drugs. And the belief is that by linking up the bench and the bedside, we'll be able to advance research more quickly. Okay, so I think I've convinced you, I hope I have, you look like you were, that research could be a direct benefit to patients, but often not. It's usually a question of benefiting other patients. And I've never ceased to be amazed by the generosity of spirit of people who find themselves with cancer, and their willingness and, in fact, desire and fervent wish to contribute to the wealth of knowledge about the disease they have. Now, of course, they want something to be done about their own cancer, but if they realise that that can't be done, then I've yet to find somebody who isn't keen to help support research efforts to make sure that somebody else will benefit in the future. So these human tissues are vital for our improved understanding of diseases and developing new treatments. And I wanted to return to the title of my talk in the last few seconds, and that was what patients can do to help themselves and research programs. Okay, there's not many slides attached to this because it's really quite simple, but yet it's quite complicated. Encourage tissue donation amongst people that you find afflicted with the same diseases. Talk to them about what it might involve. Hopefully you've now got a bit of an understanding, you probably had a good understanding before, about the benefits of material. Try and assuage their anxieties, if they have them, about the motivations of people who are working in research. I have yet to find somebody who didn't have a keen sense of the worth of their job that they're doing in cancer research, on top of being paid, on top of being famous, on top of being travelling well. They know it's a worthwhile thing to do, and their motivation, in my experience, is always very genuinely to try and help you and people in similar situations. Do take part in any debates on regulation. I did mention the Human Tissue Act briefly at the beginning. More than happy to answer questions about it that you may have. But sometimes people monopolise debates who don't necessarily take a very credible viewpoint. They assume a lot of things about people. They have a voice and they make it very loudly heard. I think in the society we live in, it depends on other people's voices also being heard. So if you've got a counter opinion to someone thinking they know best about what's good for you, then please, please, please make your voice heard. It is the strongest voice that can be heard. Someone like me saying that we need to increase tissue banking, people say, oh, well, he would do, wouldn't he, because it's going to further his research, etc., etc. People like you saying it and saying that you have a right to take part in tissue banking is much more powerful. Obviously, financial support is important. We still live in a society in England where the majority of cancer research is funded through charities rather than through the government. Unfortunately, that, of course, is under threat at the moment with various economic problems. So, the charity of your choice, please, please continue to support us in what we're trying to do. And I was asked to sort of draw your attention to the idea of having a form that you could carry around with you, I assume.
What goes with your will?
What goes with your will. Well, you might want to have it carried around with you, because by the time
you give it to your oncologist.
Right.
So, one or the other.
So just to produce this very, very simple form, it was a bit like kidney donor card in some ways, which basically very clearly declares that you want your tissues to be used for research purposes. And this sort of thing is really useful for your physicians looking after you, okay, knowing what your desires are, all right? And ethics committees and the human tissue do depend a lot on documentary evidence of people's wishes, and their default position is to say no, not to say yes. And therefore they need to have some evidence. So I think that's a really useful idea to help this area move forwards. Okay, so I've got to the end of my presentation, a little bit over time, sorry about that. Very happy to take any questions you may have.
We weren't certain whether the right place to put the form is with one's will, or to put it with one's will and inform one's oncologist, so there should be a note on your medical files that you have filled in such a form. I don't know whether you have any views on that.
Yeah. Well, as I said before, the most valuable material would be viable material. We were talking about this before. And by the time your will has been read, your cells will not be viable, sadly. So, and also, in fact, it would be too late for useful material to be obtained. So, my preferred recommendation would be that you talk to your oncologist about this at a suitable moment. Your oncologist may feel awkward about it, but I would have that conversation and say, here's my form, would you put it in my notes, please? And that's available.
I think one thing that focused our minds on this, apart from the fact that Andy Hall was speaking today, was that you may remember we talked about Ian Judson's research project. He was running into problems because he wanted to use wax block tissue samples from patients, some of whom had died. And this was the problem he was running into. Those patients who had died had not provided signed and witnessed documents to say, I give my permission. I think Ian Judson said to me a few days ago that he thinks they've worked a way around this for his project, but it is a real dilemma. There are two kinds of tissue samples. One is the living tissue, and that is something where actually you can give consent because you are alive when that tumour is removed. This needs negotiations between the surgeon and the researchers, because I was talking to some researchers from Sheffield University, and they said the tumour tissue needs to go from the patient to the lab within an hour, which means you need a motorcyclist to go from the hospital to the lab. And that's something we are beginning to work on. I think Jane has something to say about that in a few minutes. But if we're talking about wax blocks, that tissue can be stored by the hospitals for a long time. And I think what Ian is looking to do is to look at those wax blocks and follow the patient's pathway. So looking at the mutational analysis and what happened, to actually find out how those mutations actually influence the patient pathway. And this is where he's run up against real problems with the Human Tissue Act. So we have got some of these forms. I think we've got 40 forms here. You can actually download it now from the website. It is a simple form, but it does need witnessing. This is where they came unstuck. That a lot of patients have said, yes, of course you can have my tissue, but they hadn't got a witnessed document. It's a bit like a will. You've got to have a witness to make it valid. So what we're actually suggesting, the patients who are here today, take a form, sign it and get somebody else who's not related to you to witness your signature and give it to your oncologist or put it with your will and tell your oncologist you've signed it. We're not quite sure where the best place to keep the first document is, but so long as people know you've signed it, that's what we need. Because, I mean, we looked at the Human Tissue Act and we found that actually relatives cannot give permission for genetic research to be done on your sample if you're dead. It sounds daft, and the Human Tissue Act, I think, has a lot to answer for.
It's called DNA theft.
It's DNA theft. So the relatives cannot give permission, which I know the mind boggles, for some kinds of research. So please, patients, pick up a form, sign it, grab somebody you don't necessarily know to witness your signature, and put it somewhere safe. Okay?
Tell your oncologist.
And tell your oncologist you've signed it.
Can I just suggest that if you put it with your will, if you put it with your will, you get your solicitor to just give you a photocopy, and that can go to your oncologist. Yeah.
Good idea. Thank you for that. Yeah. Okay?
That's actually what we've said on the website.
Yes.
That a copy should go with the oncologist, but we felt that for a formally executed document, it probably ought to stay with the will, because that is another formally executed document. But a copy, or at least a piece of paper that says that this document exists, should be somewhere a bit more accessible.
I think any solicitor, if you take it and ask them to put it with your will, will just simply put it through the photocopy for you.
Yeah, yeah, that's fine. I think when we come to living tissues, we have a different set of problems, and I don't know how we solve these. I don't know whether you've got any ideas.
Yeah, I mean, I think the most important thing to do is to say to your oncologist, are you involved in tissue banking in your centre? If so, I'm extremely keen that material that I may come about during my treatment are contributed to that. If no, I can put you in touch with people who may be able to help through the network that you've got. And there should really be no problem with tissue being used in that way. So the majority of tissues in biobanks actually came through pathologists. So the wax blocks side of things are actually still pretty useful, not as useful as a fresh. And they sit in our pathology departments for over 30 years. After 30 years they'll be destroyed, but they're there as part of the patient record. So that material's there. So for all of you who've had a biopsy, the material's sitting in a lab somewhere, and it's a question of getting it out. Okay, and if you now sign a consent form, then that material can be released, but the pathology department need to see a copy of that. The DNA theft ping was put in because there was concerns about celebrity DNA being stolen from dustbins, and for the usual reasons that you might want to steal some celebrity's DNA, which involves paternity issues. And that's why that part of it is stuffed in there. It was a sort of, we might as well put it somewhere, we'll put it there. And the unfortunate circumstances, you've got DNA theft. The anxiety, of course, is that that your DNA tells you something about your children or your relatives. And that's why it is a bit of a fraught area from an ethics point of view. I think you had a question.
You've answered all my questions.
I've answered all your questions, I didn't know what they were.
It's about, basically the point was I wanted to make was this isn't just about, obviously, if unfortunately somebody passes on, it's about what you do now. If you're approaching the point where you're going to have an operation or a biopsy, how do people ensure that their tissues can be used for research? Because I think in some hospitals, is it not right that they haven't got a license to do anything with the tissue they've removed, and therefore as a patient, we need to kind of be a bit more proactive and rather than just assuming it will automatically be used. If you've signed the form and you've had this discussion before your operation, hopefully you can put something in place that means that tissue isn't just thrown away, it is actually sent somewhere where it can be used meaningfully.
Yeah, I think you've touched on it, but the anxiety of somebody who doesn't necessarily know the law is quite complicated. So you can hold tissues in an institution for up to 7 days without a license. My top tip to you, if you want to bamboozle your physician, is to go on the Human Tissue Act website. If you suffer from insomnia, you'll be instantly cured. And if you look for Code of Practice 9, right, 9. So if you say to your physician, well, you Code of Practice 9 of the Human Tissue Act, they'll go, whoa! Actually, Code of Practice 9 is readable. The Act itself is practically impenetrable. So the Code of Practice 9 and the one on consent, which I think from memory is Code of Practice 1, are both written in language which I'm sure everybody in this room would understand. And that gives you a bit of an itch because you know about this Act, right? And therefore, I don't encourage you to enter into a sparring competition with your physician, but if they're unsure about it, you can say, well, hang on a minute, my understanding, this is publicly available knowledge. It's your Act. You are citizens of this country. You contribute to that Act, so you have every right to kind of say, well, what about this and what about that? In the nicest possible way. You don't want to fall out with somebody who's got a big needle, do you?
Is it the case that your oncologist would automatically have access to your tissue? What about the situation where tissue removed in a general hospital and the oncologist is regionally somewhere else?
Yeah, so it can be complex in that regard. So if you present to a non-specialist unit and you have your initial biopsy taken there, so if you have an operation, the material will be in their pathology department. Okay. Now sometimes they will, if they can, they'll repeat the process, but obviously if they've got the tumour, they won't. Then they will send that material to the specialist hospital. All right. And there might be a little bit of a tussle about who can authorise it being released for research, but normally as long as the bits of paper in order, then people are quite happy to do it. Yeah.
Right. Terry, you better have a microphone. Thank you.
It occurs to me that there's already a debate about the Donor Act, whether people, when people want to donate organs. Now, surely, somewhere along the line, you can marry the two together. In other words, if you're having an operation, in theory, the NHS is going to be able to find out if anything happens to you, what you want to do with your organs. Why shouldn't that be extended so that these tissues, for example, I mean, if you're donating an organ, you've got all the logistical problems anyway of getting the organs to the right place. So it's the same thing with the tissues. Isn't there scope to tie the two together, particularly since they're debating it now?
Over to the lawyers, I think. Andy, have you got any ideas?
Well, actually the Human Tissue Authority do control organ donation as well. It's one of their responsibilities. And I think you refer to the fact that the whole business of the Human Tissue Authority is being debated at the moment as part of the arms body review. It doesn't seem like the right sort of term for human tissue, does it? And so therefore, but underlying that question might be the whole business about opting in and opting out.
Yes.
And there's, for better or worse, the English society at the moment doesn't seem to like the idea of opting out. So I think we're still with opting in. But I take your point, which is what I said about, it's like a donor card almost. Again, contact the Human Tissue Authority and ask them what they think. They're a public body, they're accountable to you as a taxpayer.